Adaptive specific host defense. So we're talking about adaptive immunity as opposed to innate immunity. Adaptive immunity is immunity that is specific for a certain pathogen. It can remember that pathogen and quickly respond to it if it sees that pathogen a second time. That's called a memory response. Adaptive immunity involves B cells and T cells. They arise just like the innate immune cells from hematopoietic stem cells in the bone marrow, but they go through a much different process of differentiation. B cells mature in the bone marrow itself. They are responsible for the production of antibodies. They're also called immunoglobulins. There's 5 different types of immunoglobulins. They're all involved in the body's defense against pathogens and toxins. And the key here is that they're involved in the extracellular environment. So antibodies don't help us for pathogens that are inside cells. B cell mediated immunity and antibodies are referred to as humoral immunity because the antibodies circulate through the blood. T cells mature in the thymus. So they're made in the bone marrow, they travel to the thymus, and then they mature. T cells are kind of a central orchestrator in both innate and adaptive immune responses, but they're primarily thought of as adaptive immunity. T cells are responsible for the destruction of cells that are infected with intracellular pathogens. They're able to specifically target and destroy in cells that are infected, and we call that cell-mediated immunity or cellular immunity. So what's an antigen? An antigen, remember, is any kind of little molecular structure on the outside of a pathogen, okay, that can be recognized as foreign by the immune system. It can be any part. A lot of times there are things you find in the surface like capsule pieces or pieces of the cell wall. Could be something like flagella, spikes on viruses, capsids, envelopes. Can also be toxins or enzymes secreted by pathogens. Okay? Antigens can be recognized by B cells or T cells, depending on the antigen. If you remember innate immunity in the pathogen-associated molecular patterns, the PAMPs were different things that are common between different pathogens. So like things that are common on the surface of bacteria or things like that. Similar function, except that what we're looking at here are very specific structures only possessed by one certain pathogen. If an antigen can stimulate a B cell to make antibodies, then it's said to be antigenic. You might hear that word. Okay. B and T cells interact with actually only tiny parts of each antigen. The parts of the antigen that the B cells and T cells interact with are called epitopes. Here are some pictures of antibodies. So most antibodies have this kind of Y shape, okay? They're made of glycoproteins. They're present in blood and tissue fluid, okay? The tips of the Y, if you will, are where the antibodies bind to their antigens. There's 5 different classes of antibody. IG for immunoglobulin, IgG, IgM, IgA, D, and E. IgG is the most abundant. It's very versatile, can penetrate into tissue. It can even cross the placental barrier. So that is this first one here that looks like a Y. IgM is essentially 5 Ys together. They are secreted from B cells during active infections. So they're a good marker for active or recent infections if they're found in the blood. IgA is commonly found in mucus. It protects the mucous membranes, can stop things from crossing over the mucous membrane by binding to it in the mucus and making it so that it can't bind to whatever its intended target is. IgD is actually attached to the surface of the B cells, acts like a B cell receptor. Okay. So when the IgD binds to a pathogen, it can tell the B cell, okay, this is the pathogen we're seeing, make a response against this. And IgE is actually important in antiparasitic defenses and can help signal to basophils and mast cells that they need to act. And it can also function in allergy, just like basophils and mast cells can. So what do antibodies do? The most important thing that antibodies do is neutralization. They bind all over the surface of pathogens or toxins. And because they're bound all over the surface, they can prevent that pathogen or that toxin from binding with its intended target. And that's called neutralization. They've essentially rendered that pathogen useless because it can't do its pathogenic thing. Okay. That process of being coated in antibody is called opsonization. So not only can it neutralize, but when a pathogen or a toxin is opsonized, it helps signal to the phagosomes of the immune system that it needs to be gobbled up and broken down. A couple more things antibodies can do. They can help with agglutination. So they can clump pathogens together by binding to the outside of them, and that makes it easier for the kidneys and the spleen to filter out. Antibodies can help activate complement cascade. Remember complement from the innate immune section. They can also help with what's called antibody-dependent cell-mediated cytotoxicity. That's a mouthful, but it's this really important process where antibodies can bind onto a pathogen and help tell an NK cell what to destroy. Or a natural killer cell. Remember, those are part of your innate immunity. So how does the immune system see pathogens inside of cells? It's a really great question, right? Antibodies are floating around in the blood and in the fluid, and they can recognize and bind to things that are out there floating around. But what about if a pathogen is inside a cell? How can we tell that a cell is infected without going inside the cell? Well, it just so happens we have a couple of very important cell surface proteins on all of our cells called MHC, or major histocompatibility complexes. The long story short, these are very complicated proteins, but the long story short is that they can hold on to tiny bits of pathogen as it's replicating inside the cell, okay? And that can signal to the immune system, because the immune system knows specifically to look for the MHC molecules. Your T cells know to look for those MHC molecules and determine if what is being held by the MHC molecule is a piece of a pathogen or just a piece of a normal host cell. Okay? MHC can display both. If it is a piece of a pathogen, then T cells can recognize that and then attack and kill that cell. So one of the other important things that MHC does is it helps the immune system distinguish self from non-self. So your specific MHC complexes on your cells in your body are very, very different from almost every other person on the planet, which is why it's so hard to find transplant matches. Because you want to find someone whose MHC molecules match as closely as yours do. Because when MHC molecules don't match, that tells the immune system that it's foreign and it should be destroyed. Another way we can see pathogens inside of cells is from antigen-presenting cells. Macrophages, dendritic cells, some B cells can ingest and kill pathogens. Okay. And then it can take pieces of that pathogen and display them on their own MHC molecules, which can then signal to T cells and other immune cells that a pathogen is present inside the body, and that can ramp up the T cell response. So speaking of T cells are specific to cells that are infected with pathogen, right? So intracellular pathogens. T cells have a T cell receptor on them that binds to the MHC molecule on either an antigen presenting cell or an infected host cell, okay? That little piece that binds to the MHC is called the T cell receptor. It's very, very specific for one specific epitope on the antigen. So the MHC is displaying a whole chunk of protein or something being made inside the cell, and that becomes the antigen. And the T cell, if it recognizes an epitope of that antigen, can then mount a T cell response. So it's very, very specific. T cells go through a very specific and very complicated process of selection inside the thymus while they're maturing to make sure that they don't recognize host cells as foreign and kill our host cells. That would be bad. And that they only recognize non-host cells. So that, and those non-host cells have to be specific for whatever pathogen the T cell receptor happens to be specific for. There are actually lots of kinds of T cells. They're all referred to by their cell surface molecules that all start with a CD. So the T cells that kill infected cells directly are called CD8 positive T cells or cytotoxic T cells. They are the ones that can directly kill. The other major subset of T cells, there are others, but we're not even going to come close to talking about them. The other major subset are CD4 T cells. So these don't directly kill infected cells, but they can help activate other either T cells or B cells to mount an immune response. So they're called helper T cells. All right, let's briefly mention superantigens. We've talked about them before. Right, superantigens are produced by some bacteria and viruses that trigger unregulated T cell activation. So they get overactivated immune system. Your immune system is in overdrive. This can result in cytokine storm. Remember, cytokines, pro-inflammatory proteins that cells make to talk to one another. They're part of the innate immune system. When the immune system is super activated, it makes way too many cytokines and just kind of overactivates everything. And that can kill someone. It's called cytokine storm. But the most important thing about T cells and B cells is their ability to remember the pathogens after they see them. So when B cells and T cells are made, they're made specific for a certain pathogen, okay? And they float around in the blood and in the lymph until they encounter that pathogen that they're specific for, okay? Some of them will never encounter the pathogen they're specific for and they go on to die. But if a B or T cell encounters their specific pathogen, they will make lots of copies of themselves, called clonal expansion. B cells will start making antibodies against that pathogen, and those antibodies will get more effective over time. So they mount this big immune response, whether it's a B cell or a T cell. And then after the pathogen has gone and been cleared from the system, the large numbers of B cells and T cells that were accumulated during that immune response, most of those will die off, but some of them will remain. And those are called memory cells, and they remember that pathogen. And if they ever see that pathogen again, they will ramp up the immune response much faster than the first time, much more effectively than the first time. And so it can oftentimes clear a secondary infection before the host even really gets infected and gets sick. That's called a secondary response. And this is the premise of vaccines. Vaccines are important because they can show our immune system a pathogen without the pathogen actually being present, so that it mounts that first primary response. A primary immune response by B cells or T cells can take days or weeks to get to its full efficacy. Okay, so if that is an actual pathogen in the system, the host of that pathogen would be very diseased by that point. So if we can use a vaccine to show an antigen to the immune system can mount a response against that antigen, and then it will carry those memory cells with it for however long those memory cells live, usually years, okay? And if that pathogen ever encounters the host, again, as a real pathogen, usually, then the host will be able to mount a quick and effective immune response and clear the pathogen before it even causes a problem. That memory response is so crucial. So that's the goal of vaccination. is to make that memory response so that if a pathogen is encountered again, after that initial response, it can be cleared fast before it causes disease, before it can get inside a host, cause infection, and replicate, and then go on to infect more people. So by making an effective immune response through vaccines, we can stop the spread of pathogens. All right, last thing we're going to talk about are different kind of classifications of adaptive immunity, because you might hear about them as you go through your career. Okay, there's natural immunity. That natural immunity can be active or passive. Active immunity is what we've just been talking about, develops after their exposure to a pathogen. Passive immunity is passed from mother to child, either in the womb or through breast milk. There's artificial active immunity through deliberate exposure like vaccines. There's artificial passive immunity. That's the transfer of antibodies by a donor into an individual. Okay. When COVID-19 first started breaking out, one of the things that scientists were talking about was using antibodies generated from people that have survived the infection and using those to treat people with an active infection. That's a really effective tool when we have yet to develop a good vaccine or good drugs against something. Okay, so patient A has had this disease, it's cleared the disease, it now has those memory B cells, it has those extra special antibodies floating around in their blood. You can take those and use those to treat someone who's infected. It's not an end-all be-all cure, but it will help that patient B's immune system to overcome the virus or whatever infection it is. Artificial passive immunity is also really common as prophylaxis against things like rabies, hepatitis A and B, chickenpox, things like that. All right, and where are we going with all of this? Adaptive immunity, memory responses, vaccines. The goal, once again, immunity in an entire population, right? In an entire population, a single individual might be susceptible to a disease, right? The vaccine might not take with them. They might not be able to mount an effective immune response. But if most of the population can mount an effective immune response, those people who are immunocompromised are much less likely to be exposed to the pathogen in the first place because it will not spread throughout the population. That's called herd immunity, and that's why vaccines are so important. Right, and I will leave you with that. Thank you.